Before I leave the lectern to Myles, let's build it up a little more. Let me say what I consider his public policy contribution. It will be very short. The practical interaction with nature has driven human civilization from the Stone Age to the global age. So far, so good. However, archaeologists, anthropologists, and historians of science and technology have regularly paid attention to the various states and developments of human-nature interactions, whereas modern political, social, and cultural historians have mostly neglected it. I harbor the illusion that this is no longer possible. Technoscience in general, and synthetic biology in particular, have become crucially important for life on this planet. Even The Economist acknowledged in 2010 that a Rubicon has been crossed. The event that triggered this statement was the manufacture of the first bacterium without a natural ancestor in May 2010. Many other Rubicon crossing events could be quoted. What we are missing is the engagement and participation of humanists and social scientists in the discussion of the new world. The ever more numerous and powerful workers in the STEM fields, science, technology, engineering, mathematics, are constructing on the other side of the river, more or less since the mid-19th century. This is a huge opportunity for philosophers, historians, and sociologists. The American Academy would be a good place for that discussion, and I'm very happy indeed that Myles is starting it.
Well, that about do it. Wine's in the foyer. No, I'm just kidding. Many thanks for the very kind introductions. Certainly start out by thanking the Robert Bosch Stiftung for supporting my research here and for supporting the American Academy. I look forward to becoming a Humboldtiana in January with the Humboldt Society. I also wanted to offer thanks to Wolf and to my fellows and my fellows' spouses and children. Much to the credit of the selection committee, the fellows have grown very close to each other. And I hope these friendships will continue once we're back on the other side of the Atlantic. I also want to give a special heartfelt thanks to the staff here. For those that come to these talks, you always hear us wax poetic about the staff, but it's very true. Certainly the kitchen staff, that was an amazing meal that some of you missed, haha. It was really great. They put a lot of time and effort into it, and we're very thankful. And I also like to thank the staff members that cater to a rather bizarre foibles, whether it's where can Myles buy his bourbon, where can Myles watch his American football, and will Myles Jackson ever meet Katrina Witt? They've done their best. So, I mean, your efforts and your efficiency are truly outstanding and unrivaled. But I think most important to me is that you people make this place a fun place to be. And don't underestimate that. You're really great, and thanks from the bottom of my heart. That does not count to my 45 minutes of the talk, by the way, which I promise, which I'll keep on time. Thank you very much for coming. I'm humbled and honored, and those were 3 great introductions, and there'll be nothing but disappointment as the result of the show. So I owe you big time. I'm going to talk about, the book is now done. It was quick. And it'll be out in stores in March and April, in stores near you. And so I wanted to give an overview of the talk. I'm not going to give a specialized talk to molecular biologists, although there's some molecular biology. I'm not going to give a specialized talk to historians. So I'm going to disappoint everybody equally by giving a very superficial talk that will hopefully tie together some interesting bits that we can talk about. The idea is that you'll get fired up about something or aggravated about something, and then we can talk about it in the discussion, and then we can drink. I've always been interested in things much more than in humans, which explains a lot, really. So I've been interested in scientific things, whether they're musical instruments, scientific instruments, whether they're achromatic lenses. In this case, I'm interested in a gene that I didn't name, it's not my fault, called CCR5. And I'm interested in objects because I like to use them as a heuristic tool as a historian to probe the boundaries between science and society. And it turns out this gene's a great gene. because it's involved in so many of the major debates that's going on in biotechnology of the late 20th and 21st century world, such as genes as commodities. Genes are patentable, and in the United States, as well as in Europe, as well as in Japan, as well as in Australia, as well as in Canada, although there is now a fight as of yesterday in Canada that we can talk about. So I'm interested in the ways in which intellectual property law has changed the conduct and content of molecular biology. I'm also interested in big pharma and their role. This particular gene, as we'll see, codes for a very important gene product, a protein. And big pharma creates small biological molecules which then will bind to the receptor of this one protein in a very important ailment. Genes or alleles, some of you might not be molecular biologists, so alleles are simply variations of a gene. If you remember Mendel back in the day, remember he had wrinkle pea, smooth pea, green pea, yellow pea, and all that fun stuff? Those are alleles. So green and yellow would be alleles for the color of a particular gene. So alleles are simply variations of a gene. And certain populations have different alleles. Part of that's due to natural selection. Darwin got that bit right. He got a lot of bits right, actually. Right? And in some cases, it's about resistance to diseases. And in this case, there's an interesting debate about the gene I'm working on and possible diseases to which it gives resistance. The next question is, precisely because different populations have alleles in different percentages, how do we consider those populations? Are they races? Right? And there has been a movement in the United States since around 2000. They're in the minority of individuals, but still, that have asked questions of, is it possible, if given enough genetic markers or bits of DNA that are unique to populations, can we figure out someone's race? It's a fascinating question, and footnote, it's not skin color. Skin color is actually a very bad proxy for race as a footnote. And there is no one genetic marker. The question is, if given 200 genetic markers, what can you say about the population from which someone hails? It's also a story about pharmacogenomics. It's the new sexy age. The idea is that we're going to cater to the individual's DNA. It explains why some person in a room will respond to a particular medication while someone will not. And it's based on the genome. So it's pharmacogenomics, and the goal, which has been one of the most important movements in medicine, I'd argue, for the past probably 20 years, personalized medicine. The magical thread that I hope ties this all together is the notion of genealogy. Given my background in philosophy many years ago, one thinks immediately of Nietzsche, so Genealogie de Morale. One also thinks of people like Foucault, who has his own version of genealogy that's based on Nietzsche, but not quite the same. And so, in the one sense, it's about the history of the present. I'm interested in where we are today and how we got there, and the fact that there were always alternatives, right? Where we are today is not natural or not inevitable, and that's what the historian will show, or at least I hope I can show towards the end of the show. Genealogy also does work. I mean, the one thing I'm not now, nor I have ever been a Foucauldian, but Foucault, I think, is absolutely right when he says that genealogy ties together the body with history in rather fascinating ways, right? So you also think of genealogy in the biological sense, because this is about a gene and tracing heredity and family trees, because that's going to be important for the people who have a special mutation of this gene. And finally, and this we're all going to do in 45 minutes or less. Finally, I swear, I'm interested. I was a historian involved in I worked on famous dead Germans in the 19th century. You can make mistakes, so people say, oh, that's no problem. But this was really interesting because now you're making decisions that had rather large repercussions. I was asked in 2009 to be an expert witness for the ACLU on a gene patented case, the breast cancer genes, BRCA1 and 2, genes of myriad genetics. I was then asked in 2012 to write an amicus curiae brief for the U.S. Supreme Court, and I was invited to the U.S. Supreme Court. It's actually a really amazing place. I mean, even I was humbled to be in that kind of world. So it's an interesting question, what can a historian do in order to play a role in public policy, which was not really clear to me when I was doing it. It became clear after a while, and I thought rather hard. What's the political context? The political context is the 25th anniversary of the fall of the Berlin Wall on Sunday. It's the post-communist world, and the question in the United States that's raging within the quarters of power of Congress that influences the work of people at the NIH and the NSF is, what should the private to public ratio of funding be? And this is in the late 1980s, early 1990s, when the Human Genome Project is getting started up. We're talking $3 billion to sequence 3 billion base pairs. It was a sale, $1 a base pair back in the day. And the question became, maybe biotech companies, private companies, should take more responsibility in the production of biomedical knowledge. And so that everyone seemed to be somewhat happy, but then interesting questions were raised, like, who owns that knowledge? Does the company own that knowledge? Or does it belong to the commons? A word that's not commonly heard in the US of A, right? I first heard it, I think, when I was in Britain. So really fascinating questions about ownership began to arise, particularly when speaking about genes. Biocapitalism, I hate that word. Everyone uses it, so I have it in inverted commas. Again, it gestures to Foucault and biopower. For me, it's more of a Marxist term. For me, I'm interested in the notion of natural entities as commodities. I'm interested in the ways in which intellectual property law structure social relationships in that world, and I'm interested in notions of alienation. The fact that people give their DNA, they certainly don't own in the means of production, and they certainly don't own in governance. Do you think of the very moving book by Rebecca Skloot on the life of Henrietta Lacks, right, who was an African-American woman from whom a tissue was taken. As a molecular biologist in a previous life, Gesundheit, I used HeLa cells. We actually thought they were from Helen Lane. We called them Helen Lane cells. It turns out it was an African-American woman from Baltimore, and it's a very sad story that was very well told in this particular case. It's a story about the United States fascinated by and interested in intellectual property and protection. You had at this time the creation of the Court of Appeals of the Federal Circuit in 1982. I think fair to say the most patent-friendly court in the world, certainly in the United States. It was also a question of ownership and protection of knowledge by 3 rather interesting players of the game. Pfizer in the biotech world, IBM in the Motion Picture Association of America. They were all upset with intellectual property theft, and they wanted to do something to change it. So they lobbied 3 groups: Congress, the World Intellectual Property Organization, and the World Trade Organization in the 1980s. And they were able to convince Congress to pass, the formation of the revisions of the Trade and Tariffs Act, 1974 and 1984, that gets changed with a Section 301 that says if you steal intellectual property, the United States has the right to punish you with trade sanctions. So they played a rather powerful role in the early 80s, and so the Federal Circuit Court of Appeal, with the Patent Office, began to extend patentability and patent eligibility. And historically, as the historian, I look back, and by and large that's true. When you have a new technology, the Patent Office is generally very pro-patent, rather patent than not patent, because the idea is that patent will encourage investment and growth, and that will help biotechnology happen. And that was certainly the ideas of the 1880s and 1880s. You can tell my 19th century story. Whoops, 1980s and 1990s in the United States. And my argument is that has come back to bite us in the gluteus maximus, that was actually a bad move. And this is an example of precisely that. One group in particular they were interested in helping out was gene sequencing companies. These are companies like Human Genome Sciences, Millennium Pharmaceuticals, and Insight. They were generally non-pharmaceutical or minorly pharmaceutical. They were interested in sequencing as many genes as possible, having very high-powered computers and computer programs, and they would license their DNA to, or access to the databanks, to pharmaceutical companies, right, for usually $125 million for a 5-year period, $25 million a shot. They did not have any therapeutic or diagnostic tools or kits. The only thing they had was gene sequencing. If they wanted venture corporate capitalist investment, they needed something to convince them that it was worth investing. That's why genes were patented, starting in 1982 in the United States. The European Patent Office and Japanese Patent Office followed shortly thereafter. This is by and large an American story. I'm also interested, I work a bit on the European story, so feel free if you have questions about the differences. Speaking to people in the European Patent Office, many of them, gene patenting is much more controversial in Europe than it is in the United States, although in the United States with the breast cancer gene it's now much better known. It was seen as a defensive strategy. The Europeans said we cannot allow all, to nuke gene patents because all of research and development will happen in the United States. As of 2013, 41% of our genes have been patented. We have about between 22 and 23,000 genes. And over 2/3 of those belong to private companies. So it's a rather important moment in history, methinks. And usually I give a talk to scientists, and they always say, but Myles, you may not patent products of nature. Well, you kind of can, and it's really a fascinating story. We've patented hormones in the United States since the first decade of the 20th century. Plants, some plants, not all plants, are patentable after the Congress passed the Plant Patent Act of 1930. Antibiotics were patented after World War II. Penicillin, by the way, was never patented as a product. The processes of invention were. Vitamins are patented now since vitamin B12, 1950s, after the 1952 Patent Reform Act. Bacteria are now patented if they're created in a laboratory, a Pseudomonas that eats oil. Oncomouse, my favorite, a poor little rodent that is genetically modified to succumb to cancer, has been patented, and of course, stem cell lines have been patented. So if you do think that patenting natural products is wrong, is bizarre, it's a really interesting philosophical question. What's the difference between artificial and natural? And who decides it? Well, the answer is the USPTO and then the various courts. And so the most important decision was made. I love this guy's name. It's the best name in the Learned Hand. I want to be Judge Learned Hand when I grow up. I mean, I'm not making that name up. He's a very famous judge. Learned Hand decided in 1911, Park Davis versus H. Mumford, the most common case used as a precedent, it's US law, so precedent cases are critical, as patenting for genes. And that is about, not about a gene, it's about a hormone, adrenaline, that was purified by Park Davis by a Japanese-American biochemist, Takamine. And the question was, is that purified substance somehow different from its natural homologue? And Learned Hand said, yeah, it is, because the patented adrenaline is purified, and that has medicinal therapeutic applications, which naturally occurring adrenaline does not have. So if it has properties which distinguish it from its natural homologue, it is patentable in the United States. And again, that's the most important case for that. What about genes? Briefly, if you're a molecular biologist, if you're not molecular, you can take a nap for the next 20 seconds, then I'll wake you up. You simply take a gene, right? You can make an mRNA copy. You then excise it, or you can excise the gene and make an mRNA copy. You make a cDNA copy from the mRNA. You put in a cloning vector, and it's now a product of human hand. My attack against that is that even though it's a product of human hand, it has the same code that it does in nature, therefore it shouldn't be patentable. But not everyone bought that argument, some did. So with that Wagnerian introduction, yep, we're fine so far. What's the story? J. Craig Venter is an infamous guy, poor guy in the United States, what Steve Chapin calls the scientist entrepreneur. He has 2 yachts and does very well for himself. And he was very upset at the National Institutes of Health, where he had been working, because of questions about patenting actually of something called express sequence tags. And so a man by the name of Wallace Steinberg, who was head of the Healthcare Investment Corporation, which was the largest health corporation in the United States in the 1990s, said, hey, Craig, I hear you're unhappy. Let's make a deal. Come to us, Institute of Genomic Research, or TIGER for short, become the director of this nonprofit organization. I'll give you $70 million. And Craig said, I want 85. So if you ever needed to negotiate for a job, J. Craig Venter's the man. He took the job, and the idea was that the Institute for Genomic Research would sequence as many genes and find as many human genes as possible. But there's a hitch. You get 85 million, there's always going to be a hitch, let me tell you. And the hitch was, he was not allowed to publish the sequences. He had to give them to Human Genome Sciences, a so-called sister, very much for-profit company, under the directorship of one of the leading virologists of our time, William Hazeltine, who was professor of virology at Harvard Medical School. Hazeltine would look at the genes. So Venter would have to give him 6 months' time, and he thought if the genes were interesting, they coded for something therapeutic or diagnostic, Hazeltine had the right to say, I want to look at this for another 12 months. So Venter was not allowed to publish the data for 18 months. That was a problem because Venter, as a part of TIGER, was part of the Human Genome Project. They were required to publish the results within 24 hours. So a very fascinating public-private dispute. The Brits were great, said, yeah, you bloody Yanks, you capitalists, we shall pull out of this if you don't get your act together. So it was really a magic moment. You gotta love the Brits, I do. One of the sequences that was sent by Venter to Hazeltine was patented. It was applied for a patent by Yi Li and Stephen Rubin, 2 scientists of human genome science. It's got a boring title. Scientists, they're not good at titles of things, but they called it the Human G Protein Chemokine Receptor HDGNR10. We're going to change the title soon, otherwise this becomes a 3-hour talk, right? And they apply for the patent on the 6th of June, 1995. They actually, as far as we know, they would never interview. Human genome scientists just never spoke to me. I feel unloved. But all of the scientists that were very angered by human genome sciences were convinced they did no molecular biology, no wet biochemistry. They determined the function of the gene by using computer algorithms, FASTA and BLAST. In the age of bioinformatics, the rather mercurial world of patenting, the argument is, let's find out by using computer homology. We get this gene, computers compare it very quickly with genes that code for known proteins, and based on a homology index, we can basically take an educated guess. It had an 81.2% sequence homology with a chemokine receptor. They then said, bingo, it's chemokine receptor. What are chemokines and the receptors? Chemokines are little molecules in the body that are important to your immune system. They tell your T cell, your white blood cells, to go to specific spots to fight infection. They're also implicated in allergic responses. They are very important molecules that are of interest to molecular biologists and universities as well as big pharma. Chemokine receptors are simply receptors, 7 transmembrane receptors on your white blood cells, which bind to the chemokines and elicit responses such as, our white blood cells are now going to attack the invader, or I'm going to give you a fever, or I'm going to make you cough and sneeze and all that fun stuff, right? Up to this point, HDGNR10 is a phenomenally uninteresting story. And you're going, what the heck did I come out tonight? About a chemokine receptor. It's about a story about bioinformatics. It's one example of a gene of literally thousands or tens of thousands up to that point. But because it's a chemokine receptor, it financially could be rather important. So the application is submitted in June. And then 3 big-time researchers at the National Institutes of Health, Robert Gallo, Paolo Russo, and Edward Berger, started making links between chemokine research and HIV/AIDS. So you now have the confluence of 2 groups. This is late. This is December. Gallo and Russo is December 95. Berger's paper is in Science, March 96. Again, this is after the submission of the patent. Things get interesting. Labs independently at the NIH, the Aaron Diamond AIDS Research Center, which is in Rockefeller University in New York, NYU, my colleagues, colleagues at Penn School of Medicine and the Dana-Farber Cancer Institute, and the Penn group was working with a group in Belgium, did experiments on this new gene, not knowing that it was that a patent had been applied on it, that this receptor, which is now called CCR5, is the HIV-1 co-receptor, which we say in English, touchdown, which has nothing to do with Landung einer Raumschiff, but literally to score a goal, it's great news. They had no idea it was an HIV co-receptor when they applied, because that was determined after the patent was already handed in. CCR5 is now a biomedical object. I said there would be no science, but I lied. There's only going to be a little bit of science. This is HIV. It has a glycoprotein. CD4 is the main receptor. This is your white blood cells, right? CD4 attaches to the glycoprotein. A stereochemical switch happens. It begins to open up. That enables CCR5 to bind. That causes the GP120 literally to go horizontal. That enables the glycoprotein 41 to make an incision in the host cell, so the genetic material of the retrovirus HIV-1 then goes into the host cell. That's classic infection HIV 101. But the plot thickens, because it turns out a year before, in 1995, a bunch of researchers worldwide, mostly at Johns Hopkins and at Harvard, realized that there were a number of self-described promiscuous gay men whose partners died of AIDS, but they never succumbed to any of the symptoms, even after a decade, right? Now, either they had amounts of HIV in their blood. One had an amount of HIV in his blood that was 3,000 times more than the amount you need to infect. That's wow, right? Some simply didn't have any HIV in the blood, and it would disappear. So that was rather interesting, and it turns out that there are a group of people who have a particular mutation of this CCR5 gene called delta 32. D for deletion, delta for deletion. And that was the biotech group of Euroscreen, which is part of Free University Belgium, the Penn group, the NYU group, and the Aaron Diamond group. And so within a month of announcing this HIV co-receptor, they said, there is this great mutation, delta 32, that if you're a homozygote, which sounds sexy, but it's not, i.e. you have a copy from mom and a copy from dad, you're by and large immune to AIDS. Ooh, touchdown. Doesn't get any better than that, right? So, Euroscreen files a patent on the CCR5 gene. They know nothing about human genome sciences. They haven't advertised it yet because it hasn't been granted, right? On the delta 32 mutation, And they cite HIV-1 recognition. I've interviewed the Belgians. They've been really great about this and said, no, we've done, and they showed me the experience. They did the wet biochemistry to show that this is the delta 32 receptor. So the wild type means normal. That's why I studied molecular and cell biology. Anytime the wild type is normal, you know it's a damn interesting subject. So this is the wild type, and it's a 7 transmembrane protein. This would be your white blood cell. This would be the exterior. AIDS binds right there at the end near the end terminus. If you have the mutation, here's the cell membrane. The receptor is on the inside of the cell. So if you think of receptor as the lock and HIV as the key, the lock is on the wrong side of the door. It can infect your cells. However, HIV can last in your blood for several, I don't know, I'm not an MD, nor do I play one on TV, but I'm also not a lawyer, nor do I play one on TV. It can last hours in the blood, and you can infect someone, right? So the moral of the story is always practice safe sex. Okay? You heard it here first. What were the responses to the patent? Wall Street had a field day. The patent was granted, right? And, you know, like I said, there's no mention of HIV in the patent. Human Genome Sciences, Inc. stock soars 50% in 2 days. They made over $1 billion, billion in a year, right? CCR5 is very much now a financial object. Biomedical researchers, not so fired up as Wall Street. So Robert Gallo said, that's like me finding another fungus. I should get all the credit for penicillin. Dan Lippman's a colleague of mine at, I hate these buttons. Dan Lippman's I'm a Luddite at heart. Dan Lippman is at NYU School of Medicine. He played one of the critical, his lab was one of the critical ones to determine the function of the gene. He said, how can you give a patent of a gene for a company that just got lucky with a computer guess and didn't do any of the work? Eric Lander is one of the leading molecular biologists in the United States. He's the director of the Broad Institute of MIT and Harvard. And he said, look, this is really dangerous that the USPTO is giving out patents is the term willy-nilly for computer sequence homology and not waiting until the molecular biologists are doing real research. In a quick attempt, because it's all about getting it in on time, turns out they got the sequence wrong in the patent. There are 1,314 nucleotides, so A, T, T, C, C, G. You do that 1,314 times, A, you go insane, B, you're going to make mistakes. There are 11 mistakes, 4 of which result into an amino acid switch, meaning the protein for which it codes. One of those was for a cysteine amino acid, this one right here. Cysteine, if you're a biochemist, you can go to sleep because you know this. If not, you can still go to sleep. I do care, but you can go. So cysteine amino acids are my favorite because they form disulfide bridges with each other, which means they fold in a particular way. If you have a different amino acid, there is no bridge. If there is no bridge folding, it folds in a totally different way. That means HIV might not recognize the protein, right? Molecular biologists are not stupid. So they took the sequence that was published in this patent specification. They had a company who can actually say, I want the following 1,314 nucleotides. Make it for me, please. I think you can get like 30 cents a nucleotide now. And they'll give you the gene back. You put it into bacteria, be expressed as a protein, isolate the protein, hybridize with HIV-1. It didn't hybridize. It's a totally different protein. Now, does that matter in the United States? No. What do you have? I don't know. Do you have it? Maybe. Congratulations, you've got a patent. It's a game anyone can play. I want to play this game when I grow up. It's really amazing. Plot really thickens. So the USPTO gets the patent on the 15th of February, 2000. That's nearly 5 years. The average amount of time that a patent clerk takes on a patent, in a biotech patent in the United States, is 17 hours. There's a huge backlog in the United States, as many as 30,000 biotech patents. That's not a lot of time to determine what's called prior art, right? So they gave the patent to HGS. They gave patents for the same gene for the same claims. Footnote, you're not supposed to do that. To Euroscreen, that's the Belgian company, in September 2002. And it turns out there's a third company called ICUS, which was a biotech company in Washington, D.C., Washington State, bought out by Eli Lilly, and they gave patents for the same gene, the same claims in 2001, again, September 2004. So the thing I love about this story, these are chemokine patents. I last checked, I think there are 19 chemokines. Everyone's patented, and these are just the first 7, because I got bored, so I just left the first 7. And it turns out 1/4 of the top-selling drugs worldwide regulate G protein receptor activity. That includes chemokine receptors. They are Claritin. I don't know if you have this in Germany. It's allergy medication, and Prozac, because it's America, it's depression, better living through chemistry, right? So these drugs are humongously important in in the world today. So the CCR5 patent really is a great example of kind of the problems that we've gotten ourselves into for being very pro-patent friendly, such as, I mean, it raises interesting questions about the natural and the artificial. What happens when you award patents to different applicants for the same gene, same claims? You can, it's America, you can sue each other. But rather than that, it's better to enter into a licensing agreement. Right? So the three did that. ICUS, Human Genome Sciences, and Euroscreen entered into a licensing agreement. So I, being a naive idiot, asked Euroscreen, so what's the deal? Like, how much money? No, we're not going to tell you that, right? Licensing agreements are only disclosed when all parties want them disclosed or if there's a lawsuit that's involved. So I have no idea how much money Human Genome Sciences made by not knowing what they had and getting it wrong. I can tell you they were bought out by GlaxoSmithKline in 2012, August, for $3 billion. So it's a good job if you can get it. What about getting the specification wrong? That turns out at the time it's now changed. It's not a problem in the United States. As long as you deposit the gene in a depository, a public depository, the American Type Culture Collection in Manassas, Virginia, there's no reason to go there unless you need to look at a gene patent. That trumps anything in the specification. So you can make errors, and people, I was told by patent examiners who would allow their names released for the book, that they actually would guesstimate that as much as 1/4 to 1/3% of gene patents actually have mistakes. But you can get, as long as it's a clerical error, you can make changes to the specification even after the patent is granted. What about basing it on computer sequence homology? 3 Nobel Prize winners wrote to the USPTO, right? Varmus, Solston, and I knew I was going to forget one of them. I'll come to me in a bit. Wrote letters saying, Watson, wrote letters saying you cannot base utility on computer analogy. There are too many sequences that are very close DNA-wise in sequence, but code for totally different proteins. The USPTO finally reneged and changed that much later in 2007. What about claims not mentioned in the patent? The fact that they didn't mention HIV, is that a problem? Do they have the rights to have all the money of anyone who wants to use that gene for diagnostics and therapeutics? The argument is they do have that right. In the US, in a composition of matter patent, you only have to name one utility. All other utilities you lock up. So if I invent a chemical and I patented the chemical and say it's a detergent, I lock up all future rights to that chemical. If you realize that this chemical is a fertilizer, this would be a bizarre chemical, you have to come to me to get the rights. I can then enter into licensing with you. So you'll get the right for use as fertilizer, but it's still my molecule because I have the composition of matter patent behind it. Interesting, Germany and France, I've been told, have limited in biotech patents to only the bits that are named in the patent. So you don't have the broad utility patent that you have in the United States. Actually very important, highly controversial, and I think a most laudable situation. Big pharma, don't worry, none is as long as this, that went away. So the genealogy now, because it's HIV/AIDS, clearly there is a story about big pharma and the lives who are HIV positive. I love this slide because I spent 5 months of my life at Caltech trying to know how they went from there to there with Francis Arnold, who's a brilliant organic chemist. And I figured it out and wrote it, and the people in the press said, "You're not even think of publishing that crap," so it went out. But this is from Pfizer Sandwich, UK. They did an amazing job. This is found through high throughput screening from one of their libraries, and then you use something known as structural activity relationship analysis, SAR analysis, in which you alter the functional groups of a molecule to get the results that you want, such that it binds to the receptor, it thwarts the competitive inhibition of the virus, doesn't cause you to die. Liver toxicity is a big problem with these kinds of drugs. And it's now called Resurrect. It's called Silzentry outside of the United States. It was produced by Pfizer. It received Food and Drug Administration approval on the 6th of August, 2007, and traditional approval on my birthday, 2008. These things are expensive. They're very expensive. In the United States, it's marketed by VIV Healthcare, which was co-sponsored, was started by Pfizer and GlaxoSmithKline. In 2009, the cost in the United States per patient was $13,767. As you might know, HIV has a very high mutation rate. Therefore, as a result of the work of David Ho, who's a great molecular biologist at the Aaron Diamond Clinic in New York, you have a cocktail of 3 drugs, which means you won't just take Resurrect, you'll take Resurrect and 2 protease inhibitors. Protease inhibitors are still the best drugs for HIV, and that will cost you in the United States just under $31,000 as of 2012. Clearly, it's made some people upset. Michael Weinstein, president of AIDS Healthcare Foundation, Greg Gonzalez, policy director of Treatment Action Group. While any new HIV/AIDS drug on the market is welcome, we urge Pfizer to use restraint and to lower the price of Zolgensma immediately upon FDA approval for this expanded use. There's a story in India with this particular drug. Natco Pharma, which makes generics, said they could produce a generic for $4,000. Zolgensma in November 2010 was just over $17,000. India has a compulsory licensing law that says if you have a patent on a drug and if it's not affordable to a majority of Indian citizens, another Indian company may make a generic for the life of the patent. So Natco applied to Pfizer. Pfizer is not convinced it can do it. It's still in the works. I'm still waiting. I actually have students whose dads work at Natco, so I get the information as quickly as I can. This is a larger story that we already know about that happened in the late 20th century, which is a fight between the World Health Organization, Doctors Without Borders, for example, and the World Trade Organization on HIV/AIDS medications in developing countries. And of course, Kofi Annan had the heart on the side of the angels to help to see what's going on. What about this Delta 32 and the notion of disease resistance and allele distribution of this magical allele that gives you, by and large, not 100%, because there are articles in the New England Journal of Medicine that show you can still be HIV positive and Delta 32 homozygous. Stephen O'Brien at the time, he's now in St. Petersburg, Russia, was at the time the National Cancer Institute, really good population geneticist. And he noticed the similarities between HIV and the Great Plague, right, the bubonic plague. Not only were they great epidemics, but they sound sexy, but it's not. Microphage cytotoxicity. The bacteria in the case of the plague, the virus in case of AIDS, attacks those cells that form your immune system. They attack the cells that are sent to attack it. So he had a rather fascinating theory that said, you know, maybe Delta 32 not only gives you, by and large, total resistance, but total resistance to HIV, but maybe also the plague historically. So he teamed up with historians and demographers and went to the little village of Eyam, England, which is the site of the Great Plague, famously of 1665, 1666. And with the demographers and historians, they went to a bunch of villages. Apparently, some people have lived in that village for 350 years. Wow, man, that's impressive. It ain't that nice of a place, I gotta tell you. But I hope there's no one from Eyam, because that's gonna be really bad. So they stay, and so they take their little cheek cells, right? They put a little agar, right? And they go away, and the scientists analyze it. 14% have this Delta 30. That's high. For an allele, that's really high. They're like, touchdown. This is Christmas all over again. It's gotta be the plague. So the idea is that this is an example of a selection factor. Think of malaria and sickle cell anemia. Sickle cell anemia is not the Negro's disease, as it was called from 1910 when it was discovered at Johns Hopkins. It's actually a disease that Italians suffer from, Greeks suffer from, Iranians suffer from, Indians suffer from. It gives you resistance to malaria. It's an environmental allele. It's not a racial allele at all. So maybe this is something going on like a selection factor. So they're good scientists. They check around the world. Scandinavians are pretty fired up. They have 16%, as high as 18%. The island is called Dagur in German, but it's an Ethiopian name, Estonian name that I can't pronounce, so I'm just going to call it Dagur, of 18%. It decreases what they call a cline to 4% in Greece and Portugal. We haven't found it in Central and Western African populations, 2% in Northern Africans, 0.1%, notice I use the square because these are not my terms, South African Africans, 9.4% of South African Caucasians, 0% among Han Chinese, and the allele is found only 0.1% in China, and the argument is due to admixture with Russia, which has such a high allelic frequency. In Americans and Caucasian Americans, again, the term that's used, about 10%, 1% is homozygous. Among African Americans, 2%, about 4 out of 10 to 1 in 2,500 are homozygous. This is a huge range of allele frequencies. Something must be going on that is selecting for the Delta 30. And I think that's a fascinating story. I get fired up about this stuff. This is a little map, and as you can see, indigenous people, again, is what they use. That clearly the allele is found where the darkest shades are in Scandinavia, the Baltic countries, and in Russia, not in places like China, not in places, southern bits of Africa, not in South America. And again, we only have it here because of the so-called European Caucasian population flow. It turns out we don't think it's smallpox, we don't think it's the plague. It turns out it's much older than we originally thought. Some people say it was smallpox. My colleagues at NYU now convinced it's staph infection. We still don't know. Something's going on. The fascinating thing to me is that it's an interesting interdisciplinary work, and I work with the people at the med school on this, between historians and molecular biologists and epidemiologists. I mean, DNA is an archive. Whether you like it or not, it is. The thing is you got to know how to read it, right? And so they're the kind of the new sexy thing in molecular biology is to collaborate with historians for the history of human migration patterns, right, throughout time, using DNA as an archive, right? So the interesting bit is, how do you describe these populations that have such a bizarre allele frequency? Are they races, right? How do we label these terms? Because that becomes rather important. In a very important article in Seminars in Immunology 2006, this is the wild type protein product. Notice how it's an interesting classification. Asians and Africans, right, which is geographic, but also Caucasians. I mean, I'm Baskin-Robbins 31 flavors, but none of my people, I think, come from the Caucasus. So, I mean, it's interesting they don't put Europeans up there, for example. This is from, this is a rather unfortunate abbreviation. It's the Proceedings of the National Academy of Sciences. Proceedings of the National Academy of Sciences of the United States of America. It would have been cut off, so chopped, so I'm not going there. So, but this is one of the most important articles, journals in the United States. I'm not making that up. And here you have CCR5 haplotypes, and now you can read it: African, pygmies versus non-pygmies, African American, Asian, Caucasian, Hispanic American. I could show you about literally scores of these. The genes in the late 90s and early 2000s were basically defined in terms of populations that were race/ethnicity. Often the scientists use those terms interchangeably in rather interesting ways. This is a story about the biology and politics of human difference. I see no twins, so if we were to take everyone's DNA, we're about 99.5% similar, right? They said 99.9 at the Human Genome Project. Turns out by 2007, 2009, we're thinking it's closer to 99.5, 99.7, right? And people were fired up at the end of the Human Genome Project 2001. They said, well, this is great because there's only one race and it's the human race. Time out, because the National Institutes of Health, together with pharmaceutical companies, decided we want to do an international haplotype map because we think that 0.5, 0.3% difference is critical. It's critical for human migration patterns, it's critical for human variation, and it's critical to understanding health. Those are my favorite SNPs, single nucleotide polymorphisms. We differ in that by every 300, 600 base pairs. Someone here might have an adenine base pair, I might have a guanine. And that explains why you respond to a particular medication, but I don't. So you can see why they're very much a study of interest for many people in biomedical research. But there are numerous ways to characterize difference. It does not have to be race. You can characterize difference in terms of geography, in terms of environment, such as the sickle cell anemia, right, and malaria. This is, if you're asleep, wake up for this, because this is actually probably the most important argument. This is very recent. There is only slightly more genetic variation between two people from different continents, say Africa and Asia, than between two people from the same continent. It sounds counterintuitive, but it's true. It means most of the variation, if you pick two people from Africa, 90% of that variation will be among those two. Compare someone with Asia with them, and that's an extra 10%. So there's not really all that much difference between populations of continents. But some argue that, but even though as small as those differences are, they're very important for understanding medicine and for understanding human migration and population studies, right? The fair copper. I can go into that if you get really confused. But deja vu all over again. Yogi Berra was a great baseball player. I think he's still alive. Was a coach for many years. Disastrous coach. And the question becomes, as a historian, is this about eugenics and Tuskegee all over again, right? It's always great when I teach eugenics in school because they always said, oh yeah, that's what the Nazis did. And then I remind them that in 1936, the Nazis actually sent a delegation to California. Sorry, Mark. Because California sterilized more Americans than all their states combined, right? That's true. So that's not, that we fumbled in the end zone, which is bad. It's like shooting an Eigentor if you don't know football. Tuskegee, you may or may not know, Tuskegee was not our finest hour. It was 600 sharecroppers in Tuskegee, Alabama, African-American males, who suffered from syphilis. Now, syphilis apparently is a fascinating disease, rumor has it. And long-term syphilis effects do really bad things to your body, there's a surprise, including heart disease, dementia. And American society, that's kind of shit, it's interesting, let's study it. So they purposely did not treat there's 600 African-American subjects for syphilis. A number of them died. It was announced in an op-ed piece in 10th of November 1972, and we all went, ruh-roh. It was rather embarrassing. So there is a sense in which the NIH, and in particular, basically saying, we messed up in the past, particularly when it comes to people of race. I mean, more people of color were sterilized in eugenics than so-called Caucasians were at the time, right? There is also a difference because the biology of today is one of the individual. It's not about the collective. I mean, it's about, I don't think, the government's going to say you have to be sterilized now. But if you are very wealthy and have very good insurance and you have, say, sickle cell anemia or say have Huntington's disease, you can go to an in vitro fertilization clinic and have various fertilized eggs and choose which one you wish to implant, right? So its decisions are now made by the individual. It's not, I mean, eugenics in the United States was not right-wing. It was the politics of progressive liberalism. It was a very paternalistic government interested in the welfare of the collective at the expense of the individual. More importantly, I do think that the biomedicine of today is about inclusion, not exclusion. And this is the work of an outstanding anthropologist in medicine, Stephen Epstein at Northwestern, who talks exactly about this. And the interesting bit is that, unlike eugenics and Tuskegee, the call for a genetics of race are by a number of biomedical researchers of color and members of Congress of color. When this was announced in 2005, I was at the meeting with a number of African-American friends, and they said, Myles, if you cannot find race at the level of the gene, this is disastrous. Because meaning the government, and I'm not the government, will want to abolish affirmative action programs. That's exactly their response. And I sat there and said, look, just because it's not in nature doesn't mean it's not real. Slavery is not based on nature, right? But it's really fascinating how the appeal to nature is so powerful that they were convinced that if it's not there, they were appalled by the notion that we were questioning whether or not race exists, which is counterintuitive. Two leading molecular biologists, Risch was at Stanford, he's now at University of California, San Francisco, outstanding biomedical program. Molecular geneticists should embrace the diversity of races since, quote, ignoring our differences even with the best intentions will ultimately lead to the disservice of those who are in the minority. Esteban Gonzalez Boytat, who is a self-described Hispanic American, also at the University of California, San Francisco School of Medicine, says, look, if biomedical investigators ignore racial and ethnic markers, we're still being prejudiced. I mean, where Tuskegee was the really horrific, egregious examples of racism, we're still being racist. Why? Because most of the samples that are used from blood banks are donated by Caucasian Americans. And that's true. I've done a lot of work with volunteer groups to go in inner cities, African Americans, Hispanic Americans, particularly African Americans saying, why should we give our blood? Think about Tuskegee. I mean, it's very much indelible in their memory, understandably so. They don't trust a lot of governmental workers, and that's where a lot of work has to be done. The argument goes, if we don't look for these genetic racial markers, then the universal symbol of medicine in America will be the white male. And that's what the FDA, that's what the FDA and the NIH talk about. Now, it's not true that the white male was always the symbol of universal symbol of healthcare. It's a rather convoluted history. And again, Stephen Epstein, does a great job in going into this. But certainly after the HIV/AIDS epidemic, there was concern because, as Epstein argues in an earlier book of his Impure Science, in the United States, HIV/AIDS was usurped by very wealthy gay men, to the exclusion of women and people of color who were suffered from HIV. We don't have very good studies, detailed studies of HIV symptoms and AIDS in women and people of color until the 1990s in the New England Journal of Medicine. That's a good 10 years after the first article came out by Michael Gottlieb in the Journal for Morbidity and Mortality. I don't know if you really want to publish in that. The Center for Disease Control, announcing a virus, announcing a syndrome that would later be called AIDS. So in the early 1990s, female congresswomen in the United States, it's been a wrong day, decide, it's like saying, I work on 19th century dead Germans, that's also kind of redundant. They decided they were very appalled by the fact that there was very little information on women for diseases. And people, the African American, the Black Caucus in Congress said, yeah, we're also upset that there aren't many studies on people of color. So the NIH passed the Revitalization Act in 1993, which says that if you receive money from the NIH for diagnostics or therapeutics, you are required to have women of color and people of color, women and people of color or women of color in your surveys. If you don't, you must give the money back. The FDA agreed. They allowed in 1993 the participation once again of women during childbearing years. They were not allowed for a while because of the thalidomide scare in Europe, which we wanted to avoid that in the United States. But now, I mean, they have to show that they're not pregnant, but women of childbearing years are in clinics, clinical trials. And the FDA requires those seeking approval from the late 90s to test the safety and efficacy of the drug in women and people of color. So you have two very powerful federal institutions, the National Institutes of Health, which is very powerful, leading biomedical research facility in the United States, and the Food and Drug Administration, saying that we need to understand human variation in terms of race. What about big pharma? The initial response, the work of Michael Montoya is wonderful on this. The initial response was, you can't do that. That's state involvement in private enterprise. Right? It's communism, not the Marx I read. But it changed its tune 6 months afterward. Because pharmacogenomics, personalized medicine, they're saying, yeah, you know what? The government's saying one size doesn't fit all. Right? We need to make other drugs. And race creates new markets for advertising. They're schlau. And in the United States, second only to New Zealand, hi Mark. The United States in 1997 became the second country in the West to allow direct-to-consumer advertising of drugs. So that you now watch American football and you see an old guy throwing a ball and it hits a tire and it doesn't go in. He then takes Levitra or some other sex potency drug, throws the ball, it goes through the hoop, his wife, his lover, whatever, gives him a hug, right? We didn't have that when I was a kid, I'm just saying, right? That happened in 1997, and there was a study by the New England Journal of Medicine 10 years afterwards, in 2007, came out in 2008, that companies that advertise their drugs on television have an increase in profit by 34.7%. So it works. There's also an intellectual property issue because if you can show that your drug has a greater efficacy in a certain race, you can renew the patent. That stymies generics. And it's happened with Bidil for African-American men, African-Americans who had a heart attack and to stop them from having a second heart attack. Amaryl was marketed for diabetes in Mexican-Americans. It turns out that there is no greater efficacy in terms of race. There are certain genetic markers that aren't quite identified with race. To show you the advertisements of big pharma, warfarin is Mercuma auf Deutsch. Mercuma, thank you. It's an anticoagulant because ethnic diversity matters when dosing with warfarin. 6th Annual Multicultural Pharmaceutical Conference, the unprecedented growth in ethnic populations across various regions of the United States opens doors to a wide array of new market opportunities for healthcare and pharmaceutical companies with the onslaught of generics, pricing battles, and direct-to-consumer competition. Reaching out effectively to America's emerging majority is a clear road to brand building and market growth. And if you ever have a conference, do not call it this, because historians will find this crap out. September 7th, 2007, Pharmaceutical Marketing Research Group, when the ivory tower goes to the ebony hood. That ain't good. That really ain't good. It was at Princeton. That makes me really wish. It's at Princeton. Right? So you have big pharma. You also have personal genomics companies. I don't understand. Americans love this crap. I don't get it. We love to find out where we're from. I'm from all over the place, right? We're all from Africa originally, unless you're from like Mars, right? So, but Americans are really fascinated about this. And so we send out to companies like 23andMe, which got in trouble, as we'll talk about, for two things. One, determine where you're from. Take that with a large grain of sodium chloride, by the way. It's not that accurate, right? They're getting better because the more people do it, the more they can actually trace lineages. But it also allows you to get an idea of any kind of alleles that might be a bit problematic. You might have certain alleles that lead to atherosclerosis or to hypertension. So you can go to your doctor, be your own expert, very neoliberal. Be your own expert. You go to the doctor and say, look, I just went to 23andMe, and I'm going to have this, I'm going to have that, I'm going to have this, I'm going to have that. What can I do, right? 23andMe got in big trouble in December. The FDA had sent cease and desist letters saying stop giving medical information on alleles until you actually show that's the case, rather than quoting studies that are not quite on. But they're a fascinating group, and this, since we're almost done, I'll play a game in case you're bored. This is a true story. Imagine an individual sends a sample of his or her epithelial cheek cells to 23andMe, and she gets the results back, and he or she, and 6 weeks later, and this person finds out that 50% of his or her DNA markers come from the Irish, from the Nile of Nine hostage. That really is a true person. A 4th century warlord who has 3 million descendants. Talk about kick-ass genes, man. That's one hell of a kick-ass gene. They're Übergenen on that now, right? What do you think? Now imagine you're in the United States and you're in a court of law and someone says, we found blood at a site and this person is 50%. What do you think this person looks like? An interesting relationship between genotype, the DNA, and phenotype, the physical manifestation. Does he look like that? I hope not. Does she look like that? I bet on Leon the Leprechaun is my favorite of all time. But did you know that it was Skip Gates, right? Who's director of the Hutchins Center for African and African American Studies at Harvard. You might know the story, I guess it was 3-4 years ago. He was arrested. He broke into his house. He was out on vacation. He didn't have the key. His driver, and he broke into his house. The next door neighbor said, 2 African-American men have broken in next. I don't know if she used the phrase African-American men. Broke in next door. The police came. He was rather upset. He was then charged with, I think, disorderly conduct, resisting arrest. The charges were subsequently dropped. It was an Obama moment. He invites him and the policeman to the White House where they have beer. And then Skip convinces the policeman to do a DNA test, 23andMe, and it turns out he's related to the Nile 9 hostage as well, so they're really distant cousins. Isn't that a wonderful feel-good story in the United States? And just go back. The Faces of America, PBS 2010, was a hit series in the United States in which he had very famous people send out their DNA to 23andMe and find out to whom they're. So he had Yo-Yo Ma, he had, I don't know famous people. That could be. She was on Desperate Housewives and she married a basketball player. Ava Longoria, thank you. She was Yo-Yo Ma was tested on Meryl Streep. And it really was a huge hit, and 23andMe really benefited from this television show. 23andMe offers CCR5 Delta 32. When speaking to the company representatives, they say it's a very sought-after test because of HIV/AIDS. It was the test of the month for family tree DNA. And it says, find out if you're resistant to HIV/AIDS, now only 199 Canadian dollars. Again, that's dangerous because you can be Delta 32 homozygous and still get it through another receptor. CCR2 is possible, right? So it's about being your own expert. It's about the 60s and 70s. There's this group as well, right? So you have the pharmaceutical companies, you have the personal genomics companies, you have the FDA, you have you also have grassroots movements who, back in the 60s and 70s fought for racial rights and also research into sickle cell anemia. Jews and Tay-Sachs was a very common motif for genetic research and grassroots movements in the 70s. Then you had gender rights, patient advocacy groups. HIV/AIDS is classic for that in the 80s and 90s. Think of all the wonderful street theater in New York, wonderful art. And the people with whom I've had the honor of working, the Breast Cancer Association, with my work with the breast cancer 1 and 2 genes. So it's fascinating to me that you have all of these various entities with very different interests coming together with a single goal in mind, which is pharmacogenomics. So to end, CCR5 is seen as a possible marker when used with others for determining race. Discussions of CCR5 was always entangled with discussions of race, so you can't ask what's politics and what's science. That makes no sense as a question, because solutions to the questions of natural order are simultaneously solutions to the question of the socio-political order in this case, right? And so the moral that you come away with, if we only understand human variation in terms of race, if that's the only lens we use, we're missing out on other important information. Again, malaria, sickle cell anemia is the classic example. If we privilege DNA as being objective, we're in danger of never addressing what I think is the real healthcare problem in the United States. That's access to medicine and your wealth, right? I mean, that's a no-brainer in my world. And genetic essentialism, which is reducing things to your genes, can be dangerous even if you're interested in it. I mean, my mom always said, Myles, the road to hell is paved with good intentions. And I never had any good intentions, but I didn't care. But it was good to know even if I did have good intentions, I'd still go to hell. So it was a win-win situation. Now for the moral to, oh man, I think I lost another bet. Sorry. What is the role, this is the last slide, I swear. The role of a historian in a controversy. So I was asked, because the people from Myriad Genetics of the breast cancer 1 and 2 genes said it's always been the case that if you have product patents, then you'll get money and research will happen. And I said I didn't think that was the case. So as a historian, I'm morally obliged, and I'm hopefully skilled at pointing out alternatives, right? So flashback. Sounds like a Cyndi Lauper song. Flashback one time. 1900. The leading country in the world that produces pharmaceuticals is, very good, Helmut gets a prize, 90%, Germany. The first unified patent law in Germany is May 1877. You're not allowed to patent chemicals and medications because, bless their hearts, they say, it's actually against the moral good, right? It's a very dangerous situation in which they can price things too high. You're allowed to patent chemical processes, and they do. Germany has the best organic chemists in the world from, I'd say, the 1830s to 1933, right? And they're able to create and be really good at coming up with new methods to create a wall of patents around a chemical. The Americans weren't so good at that, right? What we did is that we took, so the Germans would make something synthetic that would have a therapeutic response, and they would make or it would be a dye as well, right? And you patent the processes. The Americans would take a naturally occurring entity like adrenaline and isolate it and purify it, because that's what we could do best. That's why Learned Hand's decision went as it went. And we have notes. Learned Hand said, if we're going to help the American pharmaceutical company against German competition, we have to allow the patent regime in the United States to award patents for products of nature if they've been purified. It was that response, right? So it's not historically true that you must have a product patent in order to make money. It's not necessarily the case that we need to understand human variation and human differentiation in terms of race. There are a lot of lenses that one can use. And, you know, we joke around about Captain America a talk ago, but I mean, my kind of two signposts have always been Walter Benjamin, whom I don't understand, and Karl Marx. Benjamin über den Begriff der Geschichte, is the last thing he writes before he commits suicide, right? Says that histories of past victors are usually used by those in power with rather sinister aim. And in Der Erzähler, which is also a fascinating work, is a story about the history of objects. And it's about how a history of objects are an alternative history that go against a master narrative. And that's what really has always fired me up about objects. And then with Karl, it's always about the last and 18th of the Feuerbach theses, that philosophers have hitherto only interested in interpreting the world in various ways. Surely the goal is to change it. And that's what I want historians to do a little bit more of, to be at the table in times of controversy, because much was and still is at stake in terms of human health, in terms of identity. And, you know, anthropologists and sociologists have a lot to say about identity culturally that I think natural scientists can learn from, and natural scientists can also talk to them. And that's why I thank Wolf for his comment. I've been asked more often than I can even think about to give talks on what the humanities will look like in the 21st century and if you need them. And the thing that, I mean, you know, in America it's always, well, you're going to be an English major, you're going to be poor, why do you want to major in English? You know, I know how to answer that question. But I'm always shocked when I hear that question in Germany, because that's a very serious debate here in Germany, right? What is the role of die Geisteswissenschaften? Because ironically, when I'm in America, I point to Germany as being the nation in the 19th century where the Geisteswissenschaften and Naturwissenschaften were actually intricately and intimately related in very critical ways. So I've taken way too much of it, I apologize, but thank you very much for your time, appreciate it.
Microphone will be moving around, so please get your arms up and Myles, get some water.
I wish it were bourbon, but it's not.
Yeah.
Oh, my glasses. If not, we can drink now. Yep, Friedrich.
Just one question that one of the last slides brought about. Mm-hmm. Thanks, by the way, for this wonderful talk here. You pointed out the narrowing down the to sort of what you call determinism by DNA.
Right, genetic essentialism.
Yes, okay, you had a better expression. Isn't there at least one, probably more historical parallels, historical episodes where you have a similar narrowing down the focus? Think of late 19th century Robert Koch against Virchow.
That's right.
Koch claiming everything is about bacteriology, blah, blah.
Right.
And Virchow coming from a sort of social democratic side,
Right.
pointing to
Social factors.
Social factors.
Right.
Look, so and again, the constellation was there a similarity here?
Sure. I mean, there are important similarities. I mean, I've always been an anti-reductionist, so I like the similarities, right? And the irony is that the molecular biologists are, unlike that story, the critical difference is most molecular biologists are not reductionists, right? I mean, epigenetics is a very real thing. And so a lot of molecular biologists argue that we shouldn't be reducing things in terms of only genes, but about way in which genes are turned on. I mean, you have things like histones, which are proteins around which DNA. So if you're not a molecular biologist, think of histones as, what do you call the things you put thread around? Spools? The thing you put thread around, right? That's a histone. I know histones better. So sometimes, given the environment, a certain bit of the thread is exposed, that's your DNA. If you have a higher salt concentration in that particular expression, the histone is going to wind differently because of the ion concentration. A different bit of DNA is going to be expressed. Also with cytosine methylation. Cytosine methylation signals proteins to turn on these genes, but not those. And that also is an environmental factor. So I think absolutely, and the irony is, it's not the molecular biologists who are being the genetic essentialists. Often it's about, you might know the book by Nicholas Wade, which came out back in May, which is controversial. I don't think it's controversial, I just think it's wrong, right? It's been refuted by leading population geneticists, because geneticists are saying we're not that reductionist, that it's not about reduction at all. It's about and it's not even, nature versus environment. I mean, it's both simultaneously. You cannot pick a boundary. There is no boundary between the two that now this gets turned on, that gets turned. It's a very intense interaction. So I think but you're right. There are times when you have examples of people arguing for a non-reductionist enterprise. In this case, most scientists are not reductionists. A lot of people, and I'm not attacking science journalists because we have some in the audience, but I think Nicholas Wade, and I also think it's critical, right? It's an absolutely critical genre. But I think Nicholas Wade fumbled in the end zone, which is like Eigentor thing again, because he's so reductionist. And the molecular geneticists, it wasn't just the cultural anthropologists who went after him, it was the population geneticists upon whom he drew, saying, we're not making those kind of arguments. That's ridiculously genetically reductionist. Mm-hmm.
I mean, I have known molecular biologists who have been reductionists as long as they wanted to exploit their reductionist research program.
That's fair.
When they found out that it is not all of it, they changed from one day to the other and said, of course, I've never been reductionist. Right. So they have a kind of opportunistic approach to research programs, and that's fair. And that brings me to the point of whether there is not a kind of automatism going on in science. I mean, it's fair to say that there are cultural issues and political issues at stake, but they are more or less the side music of the science going on. And there is some self-correcting program going on as I said, with respect to reductionism. Yeah, and I think it's hard to say that the political context really changes the direction. It may do for a while, and of course science can go astray and can become politically abused, but the science is in itself such a strong institution in most of the developed countries that I think that we must acknowledge some kind of internalism there.
Oh, sure. I mean, not all molecular biologists were reductionists. I mean, I worked with David Baltimore, who was not a reductionist from the 1970s. Well, Strohmeyer's a couple, but he's a classic. I think he's the exception that proves the exception, right? As for the second question, I would disagree. I mean, I think you're trying to create a science that's free of culture and politics, and historically that's never been the case, I would argue, right? That solutions to the questions of the natural order are simultaneously solutions to the questions of the political order. Whether it's Boyle in stem cell research in the United States, the superconducting super collider in the United States, that was politics that determined the outcome of science. So much so that the Large Hadron Collider got to win the prize, not the United States. So I think there are various bits of, I think the relationship between science and society is complex. I do not believe that science is this sphere that's immune from it, because scientists are human, and they live in a world of today. That's not to say that they're going to go around and change, that they make it up as they go along. No, it's not to say that. So that the argument against social constructivism historically, whatever that means anymore, is that you're saying that scientists make it up as they go along. No, it's to say that they are humans, they draw upon particular skills and knowledge claims that exist in a broader collective. They are very good at communicating with each other. They're very good at things like standardization, but I would never argue that they're somehow immune from culture, which is not to argue that somehow that it makes science unobjective and wrong. Quite the contrary. I saw Adam and then, oh, Adam?
Myles, first, well, 3 quick questions.
Okay, hopefully 3
The
quick
first
answers.
the first two really, I think all the fellows and I think the staff have wanted to ask you,
Oh, no.
which is, are you fired up? Are you having fun?
Yeah and yeah.
Okay,
I think you could tell, right? I mean,
but the third question
tired,
is,
I'm okay.
the third question is there a legal pathway in the United States to making patent laws more restrictive?
yes. I mean, the one thing about the U.S. Supreme Court decision on the breast cancer genes, very good question, thanks, Adam, was the Supreme Court didn't really want to take it at first. They actually sent it back to the Federal Circuit. You appealed to the Supreme Court after the Federal Circuit. They said, look, some said we're sending it back to the Federal Circuit. And also in their deliberations, 2 judges actually said, we shouldn't have to be dealing with this because it's Congress that needs to pass something that says thou shalt not patent genes. And look, I mean, if you wait for Congress to do it in the United States, I'm getting old. It ain't gonna happen in my lifetime. So basically, the U.S. Supreme Court even said, we're in a position that we don't want to be in, right? We have the laws, and that's why when the ACLU did the case, they said it was a First Amendment problem. And then they said it's about product of nature. My job is the product of nature argument. And it worked because the US Supreme Court said this has nothing to do with Constitution, about the First Amendment. So they said, we'll take it, and we know what the existing law is about patentability, and they went back to the learned hand case and said, he said you have to make a change. When you simply take a gene and excise it from the genome, you're not making a change. It has the same genetic information as it does in nature. You can patent a cDNA copy. So it was a compromise, and I think it was a logical compromise. They also said, look, this is Congress's job, and until Congress changes it and say thou shalt or thou shalt not patent genes, the Supreme Court is the law of the land. But there is a mechanism, and it's Congress. There were attempts in the 1980s to ban gene patenting that never went very far in Congress. So that would be, does that answer your question? It would have to be through Congress that would give you the way for patenting genes. Is there Kornblum? And then 2 over there.
I think you said science is not immune from culture.
Yes, correct.
But one could argue probably more strongly that culture is immune from science.
Is or is not?
Is.
Shouldn't be.
But it is. I mean, I'm thinking now of practical examples, not
Sure.
of theoretical.
Right.
If you think of, just take the last 3-4 decades of things which have become, from science, which have become politically, culturally important. I'm sure you can find examples that I don't know about, but most of them are anecdotal. We went to the moon because the Soviets put a basketball around the Earth. I was in junior high school then. We had never heard of a space program before that. And all of a sudden, the Russians put a basketball around the Earth, and we were going to go to the moon. I mean, that's the way it happened.
No, that's right, yes. Yeah.
This genetic stuff is unbelievably interesting. But as you said, most of it is almost, at the moment at least, sort of cocktail party talk about I send in my stuff and look at I was related to a Hutu or something.
Ancestry. Personal genetics is a bit further.
And
personal
so,
medicine.
I mean, I'm overdoing it. But the point is that it doesn't, as science gets more and more and more and more sophisticated and is delving deeply into the facts, shall we say.
Becoming more esoteric.
Excuse me?
Becoming more esoteric?
Esoteric, or better. How about that?
Meh.
Culture is getting less and less able to figure out what's going on.
Yeah, I mean, this is...
Look
yeah,
at the
sorry.
debate over health care
Yes,
in the
correct.
United States, the debate over abortion in the United States.
Teaching
or
evolution in the
Or
schools.
the real killer, well, don't make me cry. No, but the real killer is climate change.
Yes, oh yeah, yeah.
Nobody has the slightest idea what's going on in my own view, but, the United Nations just told us we got 20 more years to live. You see what I'm getting at?
Yes, and
And
that's
I don't know, there's no end point to my question because I don't think there's any answer to it. But it is a big issue that science is moving far, far beyond our political and cultural ability to comprehend it.
I think the classic example is gene patenting, right? Because gene patents law takes a while to get constructed, and science moves very quickly. And indeed, some of the arguments is that you're worried about gene patenting. We're now beyond that. In a sense, we were, even when we started nuking gene patents. The answer to that, I think, is to have more people interested in public understanding of science, educational roles for scientists to talk with anthropologists and sociologists, for sociologists and anthropologists to talk to scientists, and to have an ear in Washington, D.C. I mean, have the NSF and NIH increase their budgets, because the NIH is flat. I mean, it's $32 billion, which is a lot, but it's flat-topped for, I think, 10 years now. So I think the key is that to institutionalize the ways in which it's about public understanding of science. For me, I don't care if everyone does not know that the universal gravitational constant is minus 9.8 meters per second squared. You can live without that. Yeah, that's okay. You can forget that, right? Not if you're a physicist, but you should understand the ways in which science can raise questions about ownership and authorship and identity, and that's where we need, and also questions about liberties and human origins, and that's where we need to put people who have an understanding of the humanities and social sciences together. I mean, I'm a romantic at heart, which is I'm unbelievable for most, but that's the way forward. So that when I teach a class at NYU, I teach a bunch of scientists and I make them read funny things that they never would have read before so that they can begin to connect their world with the world of humanities and social sciences. I'm less worried about the facts. I want people to understand how it is that how scientists think and how humanists think and how social scientists think. And I think the rest will fall into place after. Again, it's romantic and silly, but I actually think it's the best way forward. Yeah, Monica. Yeah, and then Monica. No, you, Monica first, and then Dan. Sorry. It's right behind you. It's right behind you, that's why.
My question is also about politics and culture and science. And this is my question. I was shocked by the slide that you showed, which I assume was generated in the United States, which showed so-called racial groups, Caucasians, people of African heritage,
That's an
whatever.
American group. Yes,
No?
It is American group. Yes, that is a group
Okay.
in the United States, correct.
So what's amazing to me about that, I mean, the United States is arguably a peculiarly racially obsessed, I don't think it's arguable, I think it's obvious, racially preoccupied society, and has been throughout its history for various interesting reasons. And what amazed me about that slide was that the categories represented there are uniquely the product of American migration racial history, right? So my question is, are there other societies who also do genomic research who argue these same things? Or is this some kind of peculiarity of the United States?
You get a prize. Thank you very much. The answer is yes, there are.
Oh, that's too bad.
No, but I have it longer if you want an explanation. Yes, there are, for interesting reasons. I mean, the interesting thing is when I talk to scientists about race, the first question I ask, well, how many are there in the world? You're not going to get a single answer, right? Because it goes anywhere from 3 to like 7. And even Hispanic Americans, according to the population census of the United States, is no longer race, it's an ethnicity. Well, that helps, right? I mean, right. So that's why part of the argument is, you should not be surprised that U.S. political categories are not inscribed in nature somehow. We're good, but we ain't that good, right? The other country that's doing this is Israel, right? And I will be there in 3 weeks to give a paper. If I don't come back, have bourbon in my honor. It's a different reason. I mean, part of this they started with Y chromosome and mitochondrial testing in the 80s. Moved to autosomal chromosome, which is the non-sex chromosome, very high technology in 2000, to trace diaspora, right? And it's an interesting story of human migration. But it is politically, clearly politically, a tinderbox, because part of the debate was, back in 2012, whether or not Jews from Northern Africa, Ethiopian Jews, were descended from biblical Israel or from a later convert. Which, you know, I don't have a horse in that race, right? I mean, I don't get it. However, you do get it if it's about a question of a right to return to the land, right? So there's a lot of work on arguing things like Jewish populations, Ashkenazi and Sephardic Jews, are closer to populations in the Middle East than they are to their European host nations, the relatively recent host nations. That Jews did not, and this is a very, very hot topic, and this is where I'm going to wear a bulletproof vest. I mean, the argument is that they are not descended from the Caucasus, from Khazar, right, where the Turks are. That's probably not true. That's also a highly inflammable topic that I'm going to run the hills for. So there are other countries that are interested in this for very, very different reasons, is the answer to your question. Dan? And then Mark. Yeah.
I have a slightly different question, although it has social ramifications too. You traced throughout your talk the kind of movement away from public funding towards privatization, and then a kind of interplay between government and business to produce jobs and industries and whatever. Can you tell us what that tendency has done to basic scientific research and the support for it?
Thank you. Ouch, that's a brilliant question. First of all, I find, and I fight a lot of people on this, so I'm probably wrong. I have problems with the notion of basic research, because basic and applied can tend to be blurred and is historically contingent. That said, most of the basic research in the United States, still by far, is publicly funded by the National Institutes of Health, without a doubt, right? What you had during the Reagan administration was a bizarre chimera of public and private being blended, right, in so many different ways. It's also true in the sciences, so that, you know, public organizations like the Bayh-Dole Act, which says that if you're federally funded, you have to patent that knowledge. It was ways in which governments could work with businesses very easily and get tax relief. Businesses get tax relief. So what you had was kind of a blending of the public and private research funds during the Reagan administration. It also continued through the Clinton administration and on. And so part of the idea, and some people have argued as a result of that, that the NIH doesn't need to have more money, that they flat-topped, and that's fine because now private research will take over and save US taxpayer dollars, right? And the NIH, I know people at the NIH who also say that they're worried that the increase in private spending or private research is that they're going to get less dollars, because the argument is, well, you can always let the corporations do that and we'll save, you know, taxpayer money. That, I think, is a very real concern. And the other concern, as I talked about, is this notion of the privatization of knowledge. We know for a fact, I bought a slide, but I don't know, that's in case you want to know. If gene patents do not kill big pharma, by the way. That's what that says. And didn't kill GMOs. Sorry, it's in German. It's in bad German. We know that patents have actually increased secrecy. So it's ironic. Gene patents do the exact opposite of what patents are supposed to do. Patents are the opposite of trade secrets, right? That's why Thomas Jefferson wrote the patent law in the United States. He didn't write that much about gene patenting, but you got to give him a pass on that one, right? So we certainly have. So 85% of biomedical, again, I'm sorry for the Germans, researchers are the opinion that patents actually destroy scientific exchange, right? Two-thirds claim that such patents actually hurt their research. 55 biomedical laboratories have actually stopped doing tests for gene tests because of patent protection. A quarter of them have been sent cease and desist letters. So that you have, with an increase in the privatization of knowledge, a lack of a sharing. And a lot of scientists have talked about, the old notion of science being this res publica literarum, where we share information. And that's the other part about the notion of alternatives. In the 1920s, Drosophilists, the best Drosophilists in the world, shared their mutants, right? I mean, it was about exchange, right, between Columbia and Caltech. So that if I gave you Drosophila, great, so if you have a leg growing out of an eye, I'll give you that mutation if you give me one that has 4 legs coming out of the thorax, right? And so there was, that's true. So there is this kind of this symbolic capital, this moral economy of knowledge sharing that is now being jeopardized because of the rise of privatization of biomedical knowledge. Does that make sense? Yeah.
But that holds true mostly for the life science.
That's what life science, correct. Yeah. Mark, and then, yep.
I think it's a question, but on the way there, I mean, it was an amazingly wonderful talk.
That's very good.
It was a real honor to listen to it. And I'm sure I don't understand, but I was struck by your answer to Adam, where the learned hand could have said, well, no, if it's already in nature, then you can't touch it, sort of thing.
That's right.
And the question has to do with that seems to force you in the position of having made a distinction between a real nature, which is found,
That's right.
and a kind of nature that's worked on, or
Yep.
and that distinction, it seems to me, echoes the sort of essentialist versus environmental. Like, in other words, science at that point becomes something environmental. If you can demonstrate that science has acted environmentally, you see what I mean? So obviously, and I want try to get to it through, because it seems to be the statement that science, more or less is going to go in a certain direction and gets deflected by politics.
Right.
Again, seems to give science itself an essentialist character,
Correct.
which then maybe suffered the environmental conditions of politics and so on. It seems to me supremely suspect narrative.
Right.
But you yourself fall into it, as we all do, when you use the word nature, and as if
There is an unsullied.
yes, there's
correct.
So the question, I'll give you the question.
I give you that.
I give you the question and try to get back to it, because what's why I learned so much is around this question of intellectual property, is of course the learned hand would have to say you don't get the patent because all you did is copy it.
Correct.
So a copy
gets the
would not be considered
Right.
an idea,
Right.
which of course as against Warhol, etc.
Right. Exactly. Right.
But of course, what's so fascinating is, in my limited high school
Oh, stop.
knowledge,
You knew
really,
cylinder.
DNA is a copying machine. So basically,
Exactly what it is.
so it's kind of, what's fascinating is you're kind of looking at the question of intellectual property around something that is in its nature, if it were to have one, copying.
Copyable, patentable.
Yeah. So the question is something like this: Do you do you think of DNA as a single object with this tiny percentage of variations through combination, mutation, environment, and whatever? Is it a thing? Let's say the most natural of things that has been subjected to this continuous variation, or do you have a kind of a different way of sort of thinking what DNA
Yeah,
is?
I cheat, because that's exactly the right question to ask. I mean, I do not have a category. I allow my actors to decide what's natural and what's artificial, right? Because the idea is, you can always make the argument that all inventions somehow have used nature in one way or another, and therefore they're not inventions, right? Certainly in the United States, as a footnote, we patent both discoveries and inventions. They're both patentable in U.S. Constitution patent law. So that makes things trickier, right? As long as you can show that your discovery has contributed something new to the art, unlike European patent law, right? Europeans are very strict about differentiating between discovery and an invention. The Americans are not. So basically, I want to see, I don't have any transcultural, transtemporal definition of nature. I would never be able to do it. There's not enough bourbon in the world to get me to answer that question. I would try, but it wouldn't be. I want to know how my actors construct the notion of nature and artificiality and why. And that's what's fascinating to me, because that's precisely, your question is precisely the problem with went on with patent law, which is it's teleological. You take patent precedents. These precedents are written not to worry about gene patenting in 19 or 2010, but to determine whether or not a hormone is patentable. That clearly we have to, as a historian, go back and ask what are the questions that they're asking about that particular case, and see how then lawyers construct their case reading back the notion of the natural and the artificial back in the archive. That's why I have to be agnostic. Does that make sense? That's how I do it. That way I'm also, because I'm a chicken, and I would never want to be able to offer a trans-temporal, trans-cultural notion of natural artificial. I actually teach a course called The Natural and the Artificial. And it's a fascinating course, precisely because there is no such thing. One is defined, it's a dialectic, one is defined with respect to the other, and it's culturally contingent.
But who is a competent actor to decide? Are the creationists competent actors?
Well, it depends. It depends what if you're talking about this, you're talking about judges, right? So you're talking about the context in which, you know, expertise is going to be asked upon. In the United States, unfortunately, creationists have way too much role in the decision of what counts as the natural. But I don't battle with them because, you know, I can only hit my head up against the wall so much before I get even more damaged. So, you know, the argument would be we look at, you know, natural versus artificial. You can determine, I mean, that was a debate about the famous automata defecating duck. Was it real or not? Well, it was Vaucanson. That was the Académie des Sciences in Paris. This is a bunch of people who are not necessarily scientists determining what's artificial. I mean, the Supreme Court judges do not have science backgrounds. Although I have to say, they did their homework, and I was really impressed with what, other than Scalia, who famously said, I don't understand any of the science, despite many of them, particularly the Nobel laureate, we had 3 Nobel Prizes, they did an amazing job explaining the science, so that people like Scalia, you know, shouldn't have written. No, I don't know what they said, but I agree with the overall opinion, and it was 9-0, which kind of upset me. But Sotomayor, Judge Sotomayor did a wonderful job in doing the homework and asking some very penetrating questions to the Myriad Genetics Council, which they could not answer. Joanna? After Bourbon?
The previous question.
Oh.
It has to do with the privatization of knowledge.
Right.
So are there also not some opposing tendencies in research going on in publicizing the knowledge using in the internet and computer to actually make it available to other scientists? Because the only knowledge I have of your field is molecular and cell biology for dummies, which I've read.
Right, very good.
And also my daughter told me that there is this internet site which is called Repository of Standard Biological Parts, which is, I think, by this guy from Stanford. And I think this has to do not with human genome, but let's say E. coli and bacterial DNA, etc.
Right.
So what these people are doing is they're taking bits of DNA and publishing it on the internet, and anybody can go there, including kids who need to learn this in high school.
Absolutely.
So is this a good direction?
I think, I mean, open source has always been, I've always been a fan of. I mean, you know, it's a very good question, and it is precisely in response to the privatization of knowledge that's going on with patents. The question then becomes, if you have something that really is very, you know, valuable, are you going to put it on the internet, right? And that's where the really important questions come. But no, I see in talking to a lot of molecular biologists, particularly at MIT has been very good about this, that they see, I mean, they also work with a lot of private companies, but there is this kind of move to publicize knowledge, make it all available. I'll give you an example in big pharma of this. When we had the haplotype map, we came up with these SNPs, these single nucleotide polymorphisms, right? The Wellcome, again, the British company said, we shall patent these SNPs. And everyone's like, what? Because we're going to patent them and we're going to make them totally available so that someone else cannot patent them, because if they try in another country, it fails non-obviousness. So you can patent. Patent does not necessarily mean you discriminate. What does patent do? Patents give you the right. It doesn't make it give you the right to do something. It gives you the right to stop other people from doing it, right? So if I have a patent on something and I want to give it away, because I'll patent it, make it universally known, and everyone else will just have to suck it up and use it and not be able to patent it. So there have been examples of where patents have been used to increase public access in the human genome. They're rare, and it turns out, why did they do this? Pharmaceutical companies were more than happy. Merck was happy to do this because they weren't interested in gaining money from SMPs. They had other areas of research that they wanted to. So it's in their financial interest to release some of the information. A lot of big pharma companies actually backed me privately saying, I'm glad you're doing this. One of the patents was destroyed in Europe because La Roche didn't want to pay the human genome sciences. So big pharma is not all big pharma are for patents, right? But no, your answer to your question is absolutely. There is a movement. The question is, will they let out something that's really, really valuable that can help bring their lab at MIT $6 or $7 million? Will they then put that on the web and make that? I hope they do, but I'm not willing to bet on that one.
I was actually curious about the methods that were used to determine or isolate the populations that were tested to see what the what the overlap in genes were and the differences. Because of course, I mean, if you look at the 19th century and the early 20th century and attempts to define race, which is what I look at, that you can see the circularity of the argument on the face of it. First, you decide who should belong to a race, and then you
test them. That's exactly right. I mean, it would differ in different areas. I mean, basically, the idea was for the haplotype map, there were rules. I mean, you had the 4 groups, the Yoruba, you had Caucasians from Utah, you had residents of Beijing and residents of Japan, right? And Troy Duster has done a wonderful article on this and said basically they were the samples that were at hand in the Coriell Institute tissue stuff. So this was absolutely perfect. And if they wanted new samples, they would have questions like, you know, are you, you know, where are you of this X race for 3 or 4 generations? And that's how they would determine what the actual race. No, there's a huge problem, as Troy Duster pointed, in the notion of collecting the data. And that's still true today. I think that's absolutely right.
Thank you for your questions, and thank you, Myles, and I think we owe it to him to let him go to his further.